So as next, we will invite Claus-Michael Lehr from Saarland, Helmholtz Institute and University. He also has a very important topic, namely the topic of crossing barriers in infectious disease, one of the challenges in clinical management of infections.
Right.
And we are looking forward to hear your progress and hopefully to use them soon in the hospital.
Yeah, that's the idea. Yeah. Good afternoon, ladies and gentlemen. Thank you very much for inviting me, Beat, to switch gear a little bit to the area of infectious diseases, which I was actually in doubt myself when I was, now it's 13, 14 years ago, invited to join the enterprise of starting an institute dedicated to infectious diseases. And there is a big, we all have been through Corona, and maybe that was not so bad yet because it was just a virus, but when we look what bacteria could arrange for us, it might be a big problem. And we have not much weapons against the bacteria, while on the other side, the number of bacteria that become resistant to anything what we have is increasing, and at the same time, the number of drugs and even of the companies that are working on this, and this has also to do with economics and that antibiotics are horribly cheap and there is not much money to make with it. But when we look at this, what actually, WHO is still predicting in 2050, I could be 90 if I make it by that time, the chance to die from any of those diseases is much bigger than to die from cancer or something like that. So, big incentive to work on it. And as industry was not doing too much, we had a reason to team up with our Department of Pharmacy from Saarland University and our National Center for Infection Research and to build the HIPS. The HIPS is the Helmholtz Institute for Pharmaceutical Research Saarland on our campus. And we hope that we can at least make some contributions. So we started in 2009 with a handful of people, but we have been nicely growing. Now it's actually, we doubled the expected size, and we started a new graduate school where we are training people to be interdisciplinary trained between clinics and pharmaceuticals. We were just expecting the first cohort to start, and I have the pleasure to coordinate the initiative, so it's quite an exciting thing. But anyway, let's come to the topic. Overcoming biological barriers and preferentially needle-free has always been my interest. So my work, while I'm a professor of pharmaceutical technology, has always been on two fronts. The one is the carriers, you could say the nanotechnology. I was pleased to learn that we can call it complex advanced delivery systems. And on the other side, in order to do that in a rational way, I thought in the beginning of my career, maybe we should use the advances of human cell and tissue culture and to try to rebuild these barriers in a meaningful way. I realize that is very difficult if you want to do organ distribution or immunology, but when we are talking essentially about the skins, the lungs, the gut, or mucus and biofilms, we can do that. And we do it human-based, and also, if you're aware of the FDA Act 2.0, these things are nowadays allowed. We are encouraged to build such something like that. It's no longer true that you must do animal experiments. And in a sense, I'm a believer that a very good human-based in vitro model could be more predictive than an animal model, which is still then showing you nice effects, but to what extent it's translating, you don't know that. So, according to this concept, I have divided this talk in two parts. I will tell you a little bit and hopefully be able to convince you that these barriers are interesting to work on, the models. And I will start with the skin. That was actually in the beginning of this millennium where people were worried about nanoparticles, that they could penetrate barriers. Silica put on the skin could end up in your brain, so there was research money and a need to investigate, and it was found out that they do penetrate. Left picture shows you nanoparticles fluorescently labeled. The right part shows you only fluorescin. The molecule is much smaller than the nanoparticle, but the nanoparticle is going into, it is even size dependent, and it's also now understood with a very plausible hypothesis that it has to do with the up and down movement of the hair that nanoparticles are really pushed. People have been worked on this from theoretical point of view. Okay, nanoparticles, please remember that do penetrate in hair follicles. If you're in doubt, do a barbecue with this nice weather, get into the coal, try later on to wash your hands, and you will see where your hairs are. It remains even dark after for a few more days. That's the proof of this concept. Pharmaceutically, you can do a few things. We started together with a colleague a few years back if we could, in a needle-free way, use that for vaccination. And indeed, we could actually show this in the so-called pig ear model, and we could show a nice follicular, humoral and cytological response with the intact skin just by rubbing the nanoparticles loaded with an antigen and some adjuvantation onto the ear. But actually, we try to work on alopecia and other things, but today, infection, there are also nasty infections of the hair follicles, diseases like acne inversa or folliculitis decalvans, where the dermatologist says you can only the surgeon help. But perhaps we can deliver the antibiotics, nano-antibiotics, by taking advantage of this phenomenon in the infected hair follicles. And what we then need is a model, and there is no model of follicular-containing human skin, and you cannot use the ex vivo models either, nor can you work successfully with the mouse. So we thought, and this is a modest attempt that I'm sharing with you, maybe we can actually use a technology by adopting hair follicles that can be kept growing in coating, in floating culture, to study drugs that are proliferative for hair growth, that we can put them into an artificial matrix that could respect or replace the tissue around it. So we use 3D printing, or we first isolated the hair follicles. We get them from cosmetic surgeons regularly. They grow, as you can see, and then we can use 3D printing to make something. I said it is simple. It's a modest approach that resembles a hair follicle, and where we were pleased to see that the hair follicles in this matrix were growing at the same time. And we could also see that actually the nanoparticles penetrate in these hair follicles in the same way, in a size-dependent way, in these artificial follicles. And we could also see that there is an infection that's according to that. So the question is if we can use that also in such a targeted way for an improved treatment. That is currently underway, maybe something for next time. But I stay with the models for a while and move on to another organ. The lungs is my favorite. I became acquainted to it when I was a postdoc in Los Angeles and met people like Judd Patton, who convinced me that this is a most attractive organ of a size like a tennis court, and the airways just like this towel. And if you really want to go to the deep air-blood barrier, then you actually have the opportunity to encounter a barrier which is thinner than an erythrocyte thick is. It's less than 1 micron in thickness over the size of a tennis court. Great opportunities to deliver drugs, of course. But what we want to have is actually a model that is relevant for that. Everybody nowadays accepts CACO2 cells as a model of the GI tract, and there is also cells of the respiratory tract with the upper airways. There is a model that's called KLU, but this is just a towel on the tennis court. If you want to go really deep to the tennis court itself, you need this squamous ultra-thin epithelium. And when we started, actually, there was just a drawing, and there was a cell line called A549, which grew in a way, but you can use that for cancer studies maybe, or toxicity, but no significant barrier. We were actually at that time the first, that's quite a few years back. Katharina Elbert, one of my first PhD students, she came back and succeeded in growing human alveolar epithelial cells in a primary culture in such a way that each of them is surrounded by a perfect belt of tight junctions and forms a measurable transepithelial resistance. After joining HIPS, it took us more than 10 years actually to successfully immortalize these cells. And there is now, since a few years, a cell line created by us, HELVI, which is human alveolar lentivirus immortalized cells. It's commercially available. It's used by more than 50 groups. It's a nice thing, but it had a little bit of problem. And the problem was the irregularity. Sorry, that was one too much. too far. This is what I wanted to show. There is now a new one, a new version that's called Arlo, which we actually created as a monoclone. And you see this is even nicer, and it also has the COVID receptor, which is of course interesting for immunological studies and for studying pulmonary infections. The deep lung is probably not where you pick up the infection, but that's the place where you eventually may die from when this corona is destroyed. We want to study infections, so we want to build more. We need something that's really called now also in regulatory context a complex model, having more than one component. And what we need actually is three components. We want the lung cells, we want the bacteria on top of it, and we want the ability to deposit aerosolized medicines and nanomedicines, and then see how the patient is doing and how the bacteria are doing. And that was very difficult because you can imagine when you put bacteria on top of cell culture, nobody reasonably does this. Within a few hours, the bacteria kill everything. But when you are careful and you study this separately, first the cells and then the biofilms, you can actually achieve a situation where you, for up to 72 hours, when you regularly apply antibiotics, keep the system in an equilibrium. And you can monitor actually how your bacteria are doing, and you see you bring down them with tobramycin, but you never eradicate them. That's exactly the situation as observed in the clinics. You can see that the host cells remain viable. You can see that the bacteria, the epithelial barrier function improves, and you can also monitor so-called virulence factors, which are important for developing novel anti-infectives, to which I come in a couple of slides. And this is what most recently is going on. We are again using here 3D bioprinting. We pre-grow the bacterial biofilms. We can write, for instance, the letters of our institutes from living bacteria, illustrating that we can make very distinct structures, grow this on top of cells, and then have the function in such way, in a very reproducible way. It works also nicely when we actually transfer that from E. coli to Pseudomonas aeruginosa. You see the same differences between planktonic and biofilmic bacteria, which is important for their study. So that was the model part. I do this not for investigating the nature. I want this to do in order to make better nanomedicines. And this is now about the carriers. When we want to make novel antibiotics that are better and can cope the infections, we cannot do just antibiotics. Bacteria will again become resistant and make it clever. What we need to do is find something different, and what we are working on at the HIPS are so-called pathoblockers. The concept is not to trying to kill the bacteria, because that makes them angry. We are trying to persuade them or just to disarm them. Or eventually you do the combination of the two phenomena, because then you can weaken them and finally kill them. And one way to realize that, for instance, is actually by a technology that Patrick Couvreur just recently had presented. You still remember Keith, I guess, a very talented student in such a European network. He went for a year or so to Paris, learned some of these things, and he made a particle that can co-deliver tobramycin as a very hydrophilic antibiotic and one of these quorum sensing inhibitors, that's the group of these novel anti-infectives, in one single nanocarrier. And the result was quite nice. My medchem friends, left slide, were already happy that when they actually could combine tobramycin, with which you can reduce the bacteria amount but never eradicate bacteria, combine this with one of these quorum sensing inhibitors at the highest concentration, at least you could eradicate it. But when we put it into the nanocarriers, let's say that's a twofold improvement. When we put it in the nanocarriers, we had a 30-fold or 50-fold improvement. That means you do achieve bacterial eradication at 50 times less of the drug that you would use by clever combination and nanotechnology. And that gave us a few nice papers, also together with Patrick, and we won a nice award. That was fine. So I think in the interest of time, maybe that one is not so important. That is a tuberculosis story, briefly mentioning what was this. When you want to make nanocarriers and bring them into the lungs, you have to cope with the problem that the nanocarriers are not ideal to be inhaled. They're actually too small. That means you must incorporate the nanoparticles into microparticles. to land and fly them down into the lungs and do the interactions. But that's all what I spent on this. I think I better go to some recent studies, also in the light of the session that we had on nanocarriers for nucleotide delivery. Since about a year, we had the opportunity to work on a new Sprint D, it should supposed to be some disruptive technologies. The ideas that one of our colleagues had would actually to transfer the principle of CRISPR-Cas, which bacteria have developed to fight phages, which are their viruses, to eukaryotic cells so that we could train them to combat our viruses. So, same thing what bacteria are doing against phages by CRISPR-Cas, you could do with human cells against other viruses. All what you need to do is actually to deliver the right thing, and this is really now multi- or polynucleotide delivery, because you have to deliver messenger RNA for the CRISPR. You cannot get the CRISPR into the cells. And you need to deliver a second nucleotide because the CRISPR needs a so-called guide RNA. So you need not only one, you need two different RNAs. And we had started a little bit before in the context of thinking about adjuvantation, where we could deliver perhaps in one single nanocarrier plasmid DNA and messenger RNA to have different expression kinetics. A very talented student from Egypt, now she is back in Alexandria as an assistant professor, she developed this system where she could do that, and looking at the results, green is plasmid, red is messenger RNA, and you can see the best commercially available transfection agents can only do one of them. With these nanocarriers, you can get green and red in the same transfection with a single nanocarrier. But this was plasmid and the other one. Another shortcoming that we learned today is the lipid nanoparticles. They are very unstable. You need cooling, and when you think about, and we had a project on this as well, applying them on the skin, they are simply too soft. You cannot rub them into the follicles. You need a solid particle, and what we came up were actually to make lipid polymer hybrid nanoparticles, where you have a solid core of polymeric nanoparticles, where you just apply a thin coating actually of the different lipids, and you can study them and you can optimize it. In a sense, it works for doing this. This was the best transfection thing, and in the context of our CRISPR-Cas system, this is the newest results, where we have some of these not solid lipid nanoparticles, but lipid polymer nanoparticles for co-delivery now of these two RNAs. Again, it's a messenger RNA for the CRISPR and the guide RNA that it needs. We can achieve both things. We can also see that we can freeze-dry them. We can see that we can nebulize them, which you all cannot do with the lipid nanoparticles as such. And I think this is maybe a new impulse for advanced carriers where you don't have to do only one nucleotide, but multinucleotide delivery. That's what I had prepared for today, and I just wanted to thank you for your attention. Thanking to my team, and also thanking for the ISAM community who had chosen Saarbrücken just a month or so ago for the annual meeting, where we had a nice meeting exclusively dedicated to pulmonary delivery. Today we had CLINAM and all the different barriers. I was very happy to be invited to make my contribution. Thank you very much.